Hi everyone. Today we have a special show and I'm excited to show you. Hello live.
What matters?
Here we are. Yep. So, welcome everybody. Today we're going to talk about brass. And I know that it's a lot of your favorite subjects when it comes to this touch trace transfer DNA. Yep, so just give me just a minute here. Okay. Now, I don't know if many of you know Clopenny. She wanted to know something I wanted to know. And I first learned about Clopenny from Get a Clue. She was doing a lot of DNA research. I hadn't talked to her until I started my channel and talking to her personally. But I really appreciate Clopenny's work. You can see here in February, Get a Clue had posted some of Clopenny's work, trace DNA and its persistence on various surfaces. Hat tip to Chloe, although I swear I highlighted this in a post long ago, but I don't want to waste my time verifying that. Make sure everyone to tell the defendant's family to avoid me like a plague. Okay, so that's the title. And so trace DNA and its persistence on various surfaces, a long-term study investigating the influence of surface type and environmental conditions. So it has been claimed that the sample in the K-bar knife is brass. I do believe multiple people have called K-bar, and it is brass. However, I do think there is a fire retardant on top of the brass to protect it, but we will get to that in a second. So this says the difficulties recovering DNA from metal surfaces is a well-established but not understood issue, which is true. This is one of the most problematic and abundant metal evidence types. Both PCR inhibition and DNA damage are thought to contribute to the unique difficulties encountered when recovering and analyzing trace DNA deposits collected from brass and other metals opposed to non-metal surfaces. The recovery of trace DNA from other metals demonstrate the unique difficulties in recovering DNA from metals. So Twitter is where a lot of my hard research and sources are, and I specifically remember posting this one. And this is particularly important because it is an ambiguous and partial trace transfer DNA profile, and it was found on a metal surface, which is the snap of the knife sheath. And these are the two most difficult factors in recovering DNA. So again, how did you do this, Moscow? It's like any question. If you dig a little deeper behind the DNA, there's an issue. So thank you, Clopenny, for opening the door to this. I first learned about it through you. I Get a Clue, and Illuminati said something about it too, which is so great. I think the more of us talk about it, the better. So here's another one that I posted. It says, science has got us where they can take qualities of antimicrobial structures, with the use of nanoparticles to make substances that were previously not known to be antimicrobial, for example, corrosive substances. This is done through metal-based antimicrobial coatings to control the spread of bacteria, fungi, and viruses via high-contact human touch surfaces that have a growing exponential rate. The antimicrobial properties of metals, such as copper, and I have a periodic table up too, just because I think they're fun. So copper is going to be abbreviated to Cu, silver is Ag, and zinc is Zn. So copper, silver, and zinc are reviewed along with the effects of combining them with titanium dioxide to create a binary or ternary contact-killing surface coatings, the self-cleaning and bacterial resistance. The snap on the K-bar sheath is most likely a fire retardant, which is a different type of antimicrobial epoxy powder paint and has similar but not the same qualities as the rest of the surfaces of the K-bar that K-bar coats. So with this black epoxy powder paint, coated carbon steel, stacked leather handle, guard. Okay, so what I'm trying to say here is that what they put on the snap is different than the other coatings that K-bar has put on their stacked leather handle, their guard, their butt, and their blade. And I will show you a little later where those objects are on that knife sheath. So here we go. Just so you get a look at the periodic table here. Here's silver, Ag, gold is Au. And what did we say brass was? It was copper and zinc. Here they are right here. Copper and zinc. So anytime you see Cu, they're talking about copper. Zn, they're talking about zinc. And so brass is an alloy of both of those. So some of these materials that they're making to be antimicrobial are a mix of multiple things. Oh, wait. You didn't get to see that, did you? I'm so sorry about that. Where did it go? Here it is. Oh, Clopenny's in the house. Hello. Sarah D. Irish is in the house. Oh, Christina Vanna, hello. Good to see you. Hello. Oh, thank you for reminding me, Clopenny. I completely forget to switch the screen. Oh, it's so nice to see all of you ladies. Thank you for visiting. So this is a special episode. We get to get really into science today. Here's this colorful, bright periodic table I found. I think you can download these if you want. They're from, yeah, look, you can do the PDF from sciencenotes.org/periodic table wallpapers. I just found this one great because it's so colorful. So here we have Cu for copper, Zn for zinc. These two alloys make brass. And I also see silver Ag, gold Au. You will see these abbreviations here, especially these ones, copper, zinc, silver, gold, as we go through some of these articles. So just picture these squares here in your mind when they're talking about those. Aww. Okay. Wow, my computer was to be slow. Okay, here we are. So we took a little detour to the periodic table to give you some of these abbreviations that they will use. Uh-oh. What? Oh, it's refreshing. See here, zinc is abbreviated as ZN, and we have copper. And so here's a little snapshot that I took from the website here. Contact killing surfaces. The global antimicrobial coatings market size was valued in US dollars at 8.3 billion in 2020 and is expected to grow by 2028. That's from 8.34 billion to 20.71 billion. That's good. Antimicrobial coatings of here's copper. What did we say? Ag was silver, sienna, zinc, and that was some sort of dioxide, another mixture, are proven to prevent frequently touched surfaces from serving as reservoirs for the spread of pathogenic microbes. They are used as antimicrobial coatings on products such as face masks, supermarket trolleys, door handles, and push bars, passenger supports in buses and trains, touch screens, and computer mice installed at public places, electronic power switchboards, toilet seats, table and chair contact surfaces. And in figure 1 here, it presents some examples of commercial products made with a copper coating to enhance their antimicrobial performance. So let me see if I can open up that. Is it this one? No. Oh, I don't know where it is. Can I zoom in on this? It didn't work, did it? Okay, but anyway, I see here the toilet seats look copper, which is weird. I've never gone into a restroom and they're copper. Okay, and this is the other article that I got, all that information I just read to you. I can open this one. Flame sprayed zinc dropped hydroxyapatite coating with antibacterial and biocompatible properties. That one is ResearchGate. So the high hydroxyapatite, short for HA, has a good biocompatibility, high strength, and low structure for toughness. So a lot of these materials, they're bashing things together at the particle level. because they need to get something that's tough and that can stand up to corrosion, and also repel. So that's why they need a low fracture toughness in this material. And the main material for artificial bones and bioceramics, because of the similar composition, that is amazing. Bioceramics, is that like glass bones? Oh my god, that's fascinating. I love that. It's been a while since I read this, so I remember some of it but not all of it. In this study, we prepared zinc dropped hydroxyapatite, I'm probably saying that wrong, by the co-precipitation method to produce artificial bone material as antibacterial properties for the use in post-joint surgery to inhibit bacterial infection or proliferation, and ultimately the incidence of osteomyelitis. my medical terms is paying off here. Okay. The co-precipitation method prepared hydroxyapatite with different concentrations of zinc chloride. The experimental results revealed that after sintering, it has some new phases generated of zinc O. Is that like zinc oxygen? And see, there's another compound there. I'm not exactly sure what that is, but they're experimenting with different materials to get different reactions. And this is fascinating that they're trying to make bioglass for bones. That is something else that's special. When we add zinc chloride, that concentration reaches 0.5 m. The minimum inhibition zone revealed the antibacterial ability of zinc HA. Increased when concentrations of zinc chloride increased to 0.125 m to 0.15 m. The granulated powder zinc dropped hydroxyapatite was coated on titanium substrate by the flame spraying method. A zinc hot coating was found with good biocompatibility and antibacterial ability in the biological and antibacterial test. So what they're doing here is they're trying to create a coating that is fire resistant, fire retardant. But in doing so, it still has to be strong enough and, well, I guess gentle enough too for these bioglass bones and, for these everyday products that we were reading about that they want to keep, like these products here. They're coating these things to stop the spread of disease and different fungi and algae and fire. So there is a coating on the snap sheath, or the sheath of the snap. Oh my gosh, Miss Potts, it's so good to see you. Hello. Welcome. We have a special episode today. Okay. What happened? Oh, am I no longer sharing? Was I sharing at all? Right. Let me show you a picture. Here you go. Right, Clopenny. Sort of like this picture I found here where the metal is pointing to this blue part. Even though the snap isn't round like this, it will still have the coating on top, and like the brass would be the blue on the bottom. Right, inside not coated. Oh, I'm so glad you caught those too, Miss Potts. So in this example, they're talking about that this is a flat oxide film on the surface of a macroscopic metal crystal. So here we go. So this was modified in order to describe oxidation of metal nanoparticles. We'll get to nanoparticles as well, with the formation of the oxide shell around the closed packed metallic core. In reality, oxidation of metal nanoparticles is often accompanied by the appearance of a hollow in a metal core, the Kirkendall effect. Herein, the analytical treatment takes this effect into account. The numerical results obtained by integration of the corresponding equation show the specifics of the oxidation kinetics in this case. And this is another reason why nanoparticles, this is another reason why DNA is difficult to abstract, because there's a chemical reaction that happens when they're putting these types of materials together, and they release metal ions. And so the metal ions that are created, they are the things that are creating issues when extracting the DNA. Oh my God, I'm still not on the page. How can I? Hold on. Oh my God, I do it every time. So sorry. Here we go. Oh, Cassidy Kane, hello and welcome. So here was what I was just reading about. For this top part here, in this example, they have a flat oxide film, which would be the orange part, and that's over the blue part, which would represent the metal. So when they place that antibacterial fire retardant on top of the brass on the knife sheath, it still has antibacterial qualities, which I'll get to that. But when you are putting these types of materials together, these nanoparticles release metal ions, and these metal ions which create issues for extracting DNA. In this review, the relation of the inorganic colloidal nanoparticles with metal ions in a biological media is discussed. Metal ions in the context of nanoparticles are relevant because of completely different reasons. First, considering unwanted effects upon corrosion dissolution. Some particular inorganic nanoparticles can release metal ions and cause toxicity, such as in the case of ZN2 plus. There's zinc. I recognize that's a zinc compound of some sort. And AG was that silver. We can go back to our periodic table in a minute. And second, intentionally modification of the nanoparticles with particular metal ions can provide contrast for imaging applications, such as GD3 ions for magnetic resonance imaging. Third, nanoparticles themselves can be used for detection of metal ions even inside cells. Thus, there are several unwanted / intended scenarios when the interplay between nanoparticles, metal ions, and biological systems is of paramount importance. And so I know people may be closed off to think, okay, DNA forensics is one thing and biomedical systems are nothing, or something different, but actually they're interrelated, because DNA is a biological material. So even when they are making ceramic glass bones, or they're talking about things that can cause cell death, we're still talking about qualities that can happen to the trace DNA, and trace DNA is usually dead skin cells. So these types of reactions are being studied, but in a different context when it actually comes to like healthcare and medical equipment. But they're still using these same practices like we learned before on toilet seats, public places, door handles, in buses, for people to stand in those areas to touch, because they want to decrease the spread of disease. And let all this layer in. I might not be going in order, but as you hear it, it'll layer on and it will start to make sense if it doesn't already. So here's another article that I was researching. So analyzing DNA from breast surfaces poses unique challenges that may result from DNA damage and PCR inhibition. To examine the relationship between metal ions present in brass, here we go. See, metal ions are in brass. They are also in the nanoparticle combinations that they use for fire retardants. So what they wanted to do was take the qualities of brass and make it so this fire retardant reacts just like brass. So even if there is a coating on the snap, it should still have those same antimicrobial qualities that kills dead skin cells. So it should have killed the DNA that is on that sheath, that knife sheath, or the snap of it. President brass ammunition. So this particular thing is talk about brass ammunition and the success of short tandem repeat profiling. 6 recovery methods were tested to determine the identity and quantity of metal ions co-recovered during DNA sampling. In addition, DNA and metal ion solutions were created at varying concentrations to determine the threshold at which deleterious effects occur. The result of this study show that copper and zinc are recovered in the highest concentrations from both fired and unfired ammunition, but at substantially lower levels than previously published. Furthermore, most metal ions co-recovered with DNA were removed during DNA purification, and complete STR profiles were generated when concentrations of copper and zinc ions were less than 0.1 m and 0.03 m respectively. So whatever they're talking about here is probably not the trace DNA. It could be like proper full profile DNA that they could get lower results than usual, but still, this is another question. Oh, thank you, Miss Potts. I'm so glad you're here too, Cassidy Kane. Oh, you guys are really, I have great subs already. Okay. Where did I go? I'm getting better at going back and forth between chat. Okay, here's this one. Recent advances in metal-based microbial coatings for high-touch surfaces. Now, this one brings up this the beer bag, because it was my understanding that viruses, beer bugs, this they not only do they have their own DNA, I do believe they can travel via DNA. Because that's like their travel mechanism, and that's how they spread. Because we know, for instance, a common cold, somebody sneezes, you have air particles. You can touch a surface that somebody previously touched, and maybe they were sick. You can touch a door handle. You know how that you can catch the common cold. The spread of our DNA is very similar to that. It's basically the same vehicle. So during the beer bug, we had antibacterial everything, the gel and all that stuff, because it does kill viruses, dead skin cells. And so this type of antibacterial killing method for the cells, it does the same thing with touch and trace DNA. So this one is called copper oxide nanoparticles induced oxidative DNA damage and cell death via copper ion mediated P38 mark activation of vascular endothelial cells. And so now, when I was talking earlier about the epoxy and the powders and how they're putting all those materials together, I don't know exactly how K-Bar does it, but across the board, this is what manufacturers are doing. They're doing something like that. And they're doing that for the purpose of these reactions to cause this cell death, to decrease the amount of virus and disease. And so what they found in healthcare, is that sometimes when you're inhaling these nanoparticles, they can cross pulmonary air-blood barrier into circulation and cause vascular endothelial injury and progression of cardiovascular disease. However, the molecular mechanism underlying the vascular toxicity of copper oxide nanoparticles remains unclear. We have recently demonstrated that the release of copper ions and accumulation of superoxide anions contributed to copper oxide nanoparticles induced cell death in human umbilical vein endothelial cells. Herein, we further demonstrate the mechanism underlying copper ions induced cell death. The methods and results, we suspended in culture medium and vigorously vortexed for several seconds before exposure. After treating the copper oxide nanoparticles, the HUVCs were collected. The cell function assays were conducted to elucidate cellular processes, including cell viability, oxidative stress, DNA damage, and cell signaling pathways. We demonstrated that the copper oxide nanoparticles uptake induced DNA damage in the HUVCs, as evidenced by YH28X foci formation and increased phosphorylation levels of ATR, ATM, P53, and H2HX. Meanwhile, we showed the copper oxide nanoparticles exposure-induced oxidative stress, indicating that the increase of cellular levels of superoxide ions, the unregulation of protein levels of the heme-oxygenase 1 and glutamate cysteine ligase modifier subunit, the evolution of levels of the malodialdehyde, but the reduction of glutamine and glutamone disulfide ratio. We also found that antioxidant N-acetate and cysteine, NAC, could ameliorate copper oxide nanoparticles induced oxidative stress and cell death. Interestingly, we demonstrated that the P38 mitogen activation protein kinase signaling pathway was activated in the copper oxide nanoparticles treated with the HUVECS, while the P38A MAPK knockdown by siRNA significantly reduced the HUVECs from the copper oxide nanoparticles, induced DNA damage and cell death. Importantly, we showed that copper ions, Schroeder tetrahemolybdate, could alleviate copper oxide nanoparticles, induced oxidative stress, DNA damage, P38MK pathway activation, and cell death. Conclusion, we demonstrated that the copper oxide nanoparticles induced oxidative DNA damage and cell death via copper ions mediated P38 mark activation in the HUVEC, suggesting that the release of copper ions was the upstream activator for copper oxide nanoparticles, induced vascular epithelial toxicity, and the copper ions chelator TMM can alleviate the copper oxide nanoparticles associated cardiovascular disease. So copper oxide nanoparticles is an important metal oxide nanomaterial which exhibits good conductive, catalytic, and antibacterial properties. It has been widely used as semiconductors, oxidation catalysts, energy storage materials, and antibacterial agents. Despite its widespread industrial and biomedical application, concerns regarding potential toxic impacts of copper oxide nanoparticles have increased rapidly. Copper oxide nanoparticles have been reported as the most toxic metal oxide nanoparticles. Oh, could this be why there's a warning on everything from California that says it'll give you cancer? I don't know. It's just like popped in my head because when you travel over to California, apparently everything gives you cancer. Oh, geez. So it's like, yeah, the stuff these types of like coatings and materials are protecting us from virus and disease, but is it protecting us from cardiovascular disease and cell death? I don't know. Ronnie, hello, welcome. I'm looking forward to it too, Clopenny. There's any way you talk about the DNA, there's a problem. And that shouldn't be. Not at all. And so, let's see. Previous in vitro studies have reported that the copper oxide nanoparticles can induce cytotoxicity, cell toxicity in numerous cell types, including human A549 lung epithelial cells, MFCF breast cancer cells, leukemia cells, HepG2 liver hepatocellular carcinoma cells, and A6 kidney epithelial cells, and rat primary brain astrocytes. Still, the molecular mechanism underlying copper oxide nanoparticles cytotoxicity remains incompletely understood. Why would they word it like that? That's so funny. Cardiovascular disease is the number one cause of death globally. Vascular endothelial cells from the inner surface of blood vessels, those dysfunction leads to the development of cardiovascular disease. Increasing evidence has indicated that engineered nanoparticles exposure may penetrate pulmonary air-blood barrier to circulation. Recently, Miller et al. Further clarified that inhaled nanoparticles selectively accumulated at sites in vascular inflammation in both animal models of disease and in human volunteers, suggesting that inhaled nanoparticles can directly interact with vascular endothelial cells, resulting in endothelial injury and acceleration of cardiovascular disease. It has been suggested that toxicity of metal oxide nanoparticles results from the release of metal ions and or nanoparticles themselves. One recent study demonstrated that autopathy-dependent release of zinc ions was involved in human A549 lung epithelial cell death and acute lung injury in mice induced by zinc oxide particles exposure. Hanagata et al. Reported that the release of copper ions rather than copper oxide nanoparticles themselves mediated copper oxide nanoparticles toxicity in A549 lung epithelial cells. Recently, we also demonstrated that lysosomal deposition of copper oxide nanoparticles facilitated copper ions release and triggered copper oxide nanoparticles induced human umbilical vein endothelial death. However, the exact roles of the copper ions and the copper oxide nanoparticles needs further investigation. Reactive oxygen species are chemically reactive molecules which mainly originate from damaged mitochondria. ROS, which is reactive oxygen species, are mainly composed of superoxide ions, hydrogen peroxide, and hydrogen free radical. Oxidative stress indicated by increased intercellular ROS is associated with nanoparticle cytotoxicity. Oxidative stress can result in oxidized nucleic acids, amino acids, carbohydrates, and lipids. Previous studies have suggested that nanoparticles-induced oxidation of nucleic acids could cause DNA damage, cell cycle arrest, and subsequent cell death. Our recent study also demonstrated that the copper oxide nanoparticles exposure caused mitochondrial dysfunction and accumulation of mitochondrial superoxide ions in HUVEX. In the current study, we clarified the potential links between the release of copper ions, oxidative stress response, and P38 MAPK activation in the copper oxide nanoparticles induced cytotoxicity in vascular endothelial cells. We demonstrated the copper ions released from the copper oxide nanoparticles induced oxidative stress in P38MK signaling activation, which subsequently caused cell damage, cell death in HUVEX. Our findings contribute to clarify the mechanisms underlying copper oxide nanoparticles toxicity in the circulation system and suggest that the chelation of copper ions may be effective means of alleviating the copper oxide toxicity. So that was a mouthful, but what this is saying is that there is a relationship here with cell death and DNA damage with the qualities of copper and also those compounds that they purposely make to have those qualities of the copper or the brass. Because because we now know that copper and zinc make up brass. So any combination of copper and zinc, they're going to have these types of qualities. And of course, they use more copper, more zinc, or a combination, depending on what kind of reaction they want out of the materials that they're making. And the human body and the human material does not react well. And that is the exact reason why they use these coatings on these public surfaces, on fire retardants, because they want to keep the spread of disease and virus low. But the issue with that is the way that we usually combat disease is antibacterial gel, or the manufacturers are using antibacterial materials. So that does also kill skin cells, because skin cells is DNA. So if you have a low number count of DNA, that antibacterial quality of whether it's a gel or the material, that is going to damage or kill the DNA. That it should have killed it. There shouldn't be, I don't think there should be any DNA on the snap. Because of these factors, like you can't, so we're at a point in science now where it's very difficult to fight against biology. And our biology apparently does not like these metal ions, these nanoparticles, these reactions that happen with those types of metals. So if our body doesn't like it and it induces cell death on a molecular level, dead skin cells are trace DNA. So those skin cells shouldn't have provided the information that they did, in my opinion. Yeah, I think this, let me see what you are doing in the chat here. Utterance. Oh, you're from Excited Utterance. So I love her. She's great. Hi, Too Fast for Fleas. I agree, Clopenny. I shouldn't have been there for that reason alone, too. See, it's just another, we looked onto another thing about the DNA, and there's an issue right there. And what was it doing for, what was that, a week or 6 days, 8 days? You said 7 days. Yeah, 7 days. It should have been gone by itself. Yes, Miss Potts. Yes, Miss Potts and Clopenny. Absolutely. You've got it. That's it. So I think that was, I just want to go over one more article for now. We can go back to this, but that was the bulk of what I wanted to say about antibacterial surfaces and coatings like enamels on brass. I think it's very interesting that these companies are saying, they want those types of surfaces to be like brass. And as I'm sure a lot of us have heard, DNA doesn't last on brass. Well, DNA shouldn't last, or will have trouble lasting on enamel as well. So this article we can go through real quick, because this one is from the National Criminal Justice Reference Service, the Office of Justice Programs. There's the National Institutes of Justice. It's from our government. So this one says persistence of touch DNA for forensic analysts. Department of Justice. National Institute of Justice. It's at MIT, that university. Yep. So they wrote this. Oh, she's from MIT. Okay. So the use of DNA evidence gathered from crime scenes to identify perpetrators, DNA profiling, is well established in the forensics community as DNA detection methods have increased in sensitivity and sophistication over the past 20 years. It has become possible to obtain DNA profiles with very low DNA concentrations, including samples collected from touched objects, i.e. touched DNA. Despite the growing importance of touched DNA evidence in criminal investigations, there are many uncertainties regarding the basic properties of touched DNA. For example, it remains unclear what biological material is actually transferred by touch. And again, this is written and supported by the National Institute of Justice, MIT, and the National Justice Reference Service, and the Department of Justice. So they're stating here that it's unclear what materials actually transferred by touch. Various studies have suggested that DNA may originate from shed keratinocytes, that's skin cells, from the outer layer of the individual's hand, nucleated epithelial cells from other fluids, or body parts that can contact with the hand, saliva, eyes, nasal fluids, from cell-free DNA reserves, either endogenous to the hands, sweat, or transferred onto the hands from aforementioned fluids. However, the relative contributions of each component to the overall sample and to its relevant properties remain unclear and likely vary by individual. Right, I think it does vary by individual because of the shutter status. Yeah, these chains of custodies, I have maintained from the beginning that they're just not going to be available. I don't think they documented anything. I don't know though. Because such DNA deposited on a surface is not visible to the eye, it is difficult to link these samples to a crime scene with the same confidence as one might a DNA sample isolated from a visible blood stain. In order to evaluate the quality of touch DNA evidence, it is therefore critically important to understand the different factors affecting how a DNA sample could have arrived at a particular crime scene. These factors include 1, the background levels of human DNA in the environment. 2, the dynamics by which DNA may be transferred to a surface by touch or other relevant activities. 3, the persistence of DNA samples under different environmental conditions. A handful of studies have investigated how factors such as time, surface type, and environmental conditions, i.e., temperature, humidity, ultraviolet light, affect the quantity and stability of touch DNA. The studies generally focus on a specific scenario, particularly surface type or simulated crime-related activity, and involve relatively small sample sizes with limited or no statistical analysis. The main studies make it difficult to draw generalizable conclusions. Another factor confounding quantitative studies of touch DNA persistence is, in fact, the amount of DNA deposited by touch varies significantly between donors, right, because of the shedding status. Several studies have circumvented this challenge by depositing controlled amounts of purified DNA or cell suspensions on surfaces. However, it is unclear whether the stability of actual touch DNA samples is similar to that of purified DNA or cell suspensions. There is therefore a need for comprehensive, well-controlled studies that provide practical data about the basic properties of touch DNA evidence in real-world experiments to understand how long touch DNA sample would be expected to persist in a particular environment. The current study was designed to address this need. Right. They don't know. Science does not know how long. Granted, it's not long, but we still don't know when or how these transfer DNA samples get somewhere. So, touch DNA collected from donors and commercially purchased through DNA were both deposited on several representative surface types. The impacts of temperature, humidity, ultraviolet rays, radiation, and exposure was determined by the DNA quantity and quality. DNA persistence was addressed by several metrics, including 1, the total DNA concentrations as measured by quantitative polymerase chain reaction, 2, degradation index as determined by ability to amplify longer DNA targets, and 3, suitability for DNA typing analysis by short tandem repeat. Summary of literature on persistence of touch DNA. Sample time. Whoa. What is this? Collected activity by volunteer by scratching. Tested objects, fingernails. 60 volunteers. 3 samples from each participant. Up to 8 hours. Whoa. So does that mean touch DNA under fingernails? Uh-oh, what did I do? I pressed the button. Hold on, guys. Oh, hello, American Princess. Welcome. Okay. So this one, sampling time. Controlled use by volunteer on knives. 3 replicated per conditions, up to 1 week. Huh. Controlled activity by volunteer, grab and struggle model. Cotton polyester blends, maybe like a shirt or clothing. They took it from 4 volunteers and they used 3 fabrics per conditions, and that lasted up for 7 days. Whoa! This is a great graph. Sampling time, surface type. Glass, metal, paper, plastic. 6 volunteers, up to 40 days. Okay. Blood, fingerprints, stains, and hair. No touch DNA, aluminum paper. Oh, that doesn't give me anything now. What do we have here? Envelopes, cotton fabric, test tube, plastic tube. Glass slides. Up to 6 weeks. So they do have expiration dates. And remember, this one's from MIT, the Department of Justice, National Institutes of Justice, and they're the ones who put out these times. Although up here it says metal, I think that we've just figured out that metal was only a few hours, not 40 days. Here we go. Brass, aluminum, nickel, glass, tape, wood, up to 8 days. Well, at least they have some times on here, but still, it's kind of like it's not completely hammered down yet. Hi, American Princess. So I know that there were other articles out there that said brass lasts up to 8 hours, and also on this graph we were just looking at, it says sampling time, controlled activity by volunteers, scratching. So I guess that would be DNA under the fingernails. We don't have much. Maybe it'll tell us more about this study down further, but it says under the fingernails it said 8 hours. How do surface type, environmental conditions, and exposure time affect the stability of touch DNA evidence? Does the stability of touch DNA deposits differ from controlled DNA deposits? So, for example, involving touch DNA, volunteers were asked to handle stainless steel bolts, cotton squares. The bolts were selected as a representative smooth metal surface that was easy to handle, could be cleaned, and could be obtained in large quantities. Cotton fabric was selected as a representative rough porous surface. Photographs of substrates, bolts, and fabric were cleaned prior to minimize background DNA contamination. Bolts were washed in sequential baths with 10% bleach and distilled water. Cotton squares were exposed to ultraviolet wavelength light for 20 minutes. Donors were instructed to avoid washing hands within 10 minutes of handling objects. Handle objects in sets of 4 with at least 30 minutes in between rounds. 3, handle each object for approximately 10 seconds in either their left or right hand. And formal randomization scheme was used to account for a round order of handling within a round, and use the left versus right when distributing objects amongst experimental conditions. After donors deposited touch DNA, the objects were stored in opaque boxes overnight, the ambient temperature, and were used for experiments within 24 hours of handling. Okay, so here they were experimenting with low temperature and low humidity. Collectively, these studies provide the most comprehensive information to date regarding basic properties of touch DNA evidence and enable improved recommendations to be made to the forensic science community regarding best practices for their interpretation and evaluation of touch DNA evidence. These studies also generated a number of recommendations for the best practices when working with touch DNA samples in a laboratory setting. So that reminds me, we were watching yesterday. What's his name? Vsauce. He was telling us about the dumbest math, which is probability, and how the likelihood ratio is very confusing, and the prosecutor's fallacy, how the prosecutor will confuse two different types of statistics, in which it could act, the DNA could actually be exculpatory to the defendant. And sometimes if that happens, and Bishop Barlow had said, yes, Bill Thompson is essentially using the prosecutor's fallacy. That is an excellent point, Ms. Potts. And so with current science today, it seems like the FBI, law enforcement, they're completely content on how they've been doing things. Some of the public is completely content on believing, okay, DNA works like it does on CSI, so I don't need to worry or learn about it anymore. But the problem is science has gone beyond law enforcement's current understanding and the public's current understanding. And until we address those types of things, we're going to continue to see all the same mistakes, wrongful convictions, mistaken identities, and I guess it's very hard for an industry or an environment like law enforcement to actually understand what they're doing and change their ways, because of course they think that they're the expert on what they're doing, which they are in some aspects, but not when it comes to DNA. Whenever I read these scholarly journals, sometimes I can feel the frustration in these scientists that, or these scholars, that, sometimes police are working blindly. They just pick some DNA sample anywhere from the scene, and then they just go with that. And it's like, well, you should, there was an article that I was reading that there's different strategies on how to locate better locations for DNA. But it's going to be really hard to change an industry that doesn't want to change. And then we were reading yesterday Inside the Cell by Bishop Barlow, and she was saying that there's issues with these databases, that these databases are showing search results or hits or that are unknown, and they need to be researched and looked into, because now that we have a database, anytime you put information in a database, it's always going to change depending on how the database works, what it's looking for, how it returns things. Are these things actually related? What's going on? So you really have to look at all the way around, what you're putting in, what you're getting out, the different functions, how they're pairing things. And so Bishop Barlow says there was an issue with these database searches, but the FBI doesn't want to put scientific experts on the job to figure out why these anomalies are happening, and these anomalies for random matches, that they're not understanding why more and more people's DNA are actually matching. And that definitely needs to be addressed, because if more and more people's DNA are matching coincidentally, then that likelihood ratio is completely wrong, just completely wrong. And I think that's what they're finding, is because more and more people are being added to this database. Our DNA, our criminals' DNA, or anywhere really, Ancestry, it's all being cataloged. So whoever does have access to that information, they're going to be able to see patterns that normal people can't. And so when you see patterns like that, we do have to study these things so that we can find these patterns, and so we can learn from them. Because if we don't address these database issues, we're going to get these wrongfully convicted, and it's going to be continually making the same mistakes. And that's so sad. Like, I know that as a society, for one, we can be better. We can have higher scientific standards. Two, we can learn as we go. Sometimes this DNA stuff is like really fun to learn, and that kind of makes us better jurors, that, we have a clear and fair understanding of what's going on in this DNA world. Well, hello, welcome. Ink Masquerade. Welcome. So that's what I had today. If you guys want to go over brass or how antibacterial surfaces and materials and gels, also kill trace DNA or brass again, let me know. If you want me to go over an article or talk about a certain subject, put it in the comment section or contact me on Twitter. Thank you all for visiting me today. I had lots of fun. And I hope you all enjoy the rest of your weekend. All right, take care. Have a great day. Bye, everyone.